Okay, so we're going to talk about bacteriological monitoring. Already kind of talked about our organization and about Global Water Watch that we're a part of. This is an example of a bacteriological monitoring workshop that Global Water Watch was doing in Kenya. We've already covered why is it important to monitor water. A couple of the good reasons are, is it good habitat for wildlife, and is it safe for people? And finally, is it getting better or worse, and why? So we know that water quality is important for salmon and other things that live in the creeks, and things that depend on those salmon. Water quality is also important for people. We're lucky that our drinking water quality around here is good, but from a recreational standpoint, a lot of times people are not aware of the impact that water quality could have. And so in particular, bacteriological water contamination is something that people don't really think about, but people recreationally access little streams, like kids go play in the streams, where the stream may actually not necessarily be safe for them to swim in or play in. This is an example again of Little Swamp Creek when the water quality wasn't so great and we had the high bacteria level. Let's talk about bacteria. They're single-celled organisms. You can't see them. They're all over the place. They're on us. They're inside of us. They're necessary to a lot of processes in life. Essentially, they help break things down and transform things from one form of matter to another. And so they're integral to life. They come in different families. So the ones that we're mostly concerned with are a type of coliform bacteria, and in particular E. coli. But you'll see that the sampling that we do is for, in particular, it tests for E. coli and other coliforms, and the color coding on that tree sort of represents what you'll see with the color coding, the color that the colonies show up in our bacteria tests. So E. coli is the thing that we're testing for. E. coli is indicative of fecal contamination, but it isn't necessarily 100% certain that it means fecal contamination. So when we see high E. coli, it indicates likely high fecal contamination, but it isn't necessarily synonymous with that. Around the world, this kind of contamination of water is a major problem, and which is why a lot of countries around the world have unsafe drinking water. In countries like Haiti, where I've done some volunteer work, they don't have sewage treatment in a lot of areas. And so, for instance, they were dealing with a big cholera outbreak there. The main thing that we're checking for around here is a particular variety of E. coli, and that's the variety that can make you sick. Here's an example of where E. coli was traced to a water park, and I think I've got a little video.
A popular body of water in Shelby County is filled with dirty water.
The Cahaba Riverkeeper recently discovered high levels of E. coli in Buck Creek. Tonight, ABC 3340's Honora Gathings is fighting for your health. She is live in Helena at Buck Creek. And Honora, is it safe to swim there?
Well, Dave and Brenda, the executive director of the Cahaba Riverkeeper, says a dip in this water could make you sick. There are no federal or state agencies regulating Alabama's creeks, and certainly no signs warning about dirty water before you take the plunge.
Janet Hernandez and her 4-year-old son often skip rocks in Buck Creek, but they have a rule: feet and only feet first.
It's just our feet. I would freak a bit more if he started swimming in it.
Other families don't hesitate to wade on in for fishing, swimming, or tubing, but Hernandez,
To swim in it, I don't trust it enough.
less than a foot from the family, a layer of gunk floated near some rocks. We sent the executive director of the Cahaba Riverkeeper a picture. Dr. Meyer Crawford believes it's either algae or scum. She says neither one is good.
Very few people are doing any kind of testing.
The Cahaba River Keeper started testing 8 points along the Cahaba River last summer, starting at Buck Creek and ending at Trustville Springs.
No one else is doing it. I think it's pretty important.
The results were documented and photographed. The dark spots are E. coli. Crawford says unsafe levels were found in Buck Creek multiple times, but an April 14th sample set a new high.
It's called too numerous to count. You can't even count the number of little dots.
Crawford says any E. coli can make you sick.
You don't even have to drink it if you have an open cut. If you're just waiting and you have an open cut on your leg, or you could become infected that way too.
E. coli comes from feces, but no one is certain what is polluting Buck Creek. The EPA is monitoring 3 companies upstream of Helena for toxic releases and more than 100 others for water discharge. But there are no current violations. The Alabama Department of Environmental Management says it isn't aware of any either. Helena's wastewater treatment plant is just over the Buck Creek Dam, about 1/4 of a mile upstream, but it is also in compliance and closely monitoring its discharge.
We're going to find out, I hope.
But until then,
I'd rather take the kids to the pool or something where it's really clean. They can actually swim.
Now this is a look at the substance that we sent a picture of to the Cahaba River Keeper. In less than a month, the River Keeper will resume testing of this water. The focus, though, will be over this dam upstream to help identify the source of the E. coli. The River Keeper will post the results to his Facebook page every Friday. Fighting for you in Helena, Anwar Gathings, ABC 3340.
And if
So I thought I'd just check in and see if you guys have any reactions to that or any thoughts about that contamination issue. Something that we have around here as well. So when you learn about, we'll talk about some of the things that creeks are so-called listed for, but creeks can be listed for different pollutants for having known too high levels of different pollutants, and fecal coliform is actually a pretty common one around here. So for instance, Swamp Creek, we're in the Swamp Creek watershed, has a plan and a listing for too high of level of that. So it's pretty common around here, unfortunately. So where does it come from? We talked about pets is one place where it can come from. This is a relatively recent article last year, I guess, in Kirkland. Kirkland did an interesting study. So they were, this was, I think, at Juanita Beach Park. They did a study where they had volunteers go around and plant little flags everywhere that they saw dog poop. And they were incentivizing people to pick up dog poop. And they were also documenting how much dog poop there was. And so basically what they found was 2 things. 1, there was a huge amount of dog poop being deposited at this park. And 2, by pointing it out with these flags and talking to people about it, they were actually able to get people to change their behavior and reduce the amount of dog poop that was being left in the park. Another place that bacteria can come from into streams and water bodies is from these things called side sewers. So people's homes are connected to the main sewer line through this side sewer, and these things can occasionally leak or actually get improperly connected. One of the groups that we were working with is called the Thornton Creek Alliance, and Thornton Creek has very high levels of bacteria. It's one of the major watersheds in Seattle. And so Thornton Creek Alliance did a citizen science project in conjunction with Seattle Public Utilities, where they went out essentially weekly with volunteers and monitored multiple sites throughout the watershed for bacteriological contamination. And ultimately they found, when they compared the results all around the watershed, they found some areas that were really high. And when they did some more, they narrowed that testing in, they found essentially a apartment complex that had an improperly plumbed sewer line that had been plumbed into the storm sewer. So it was basically putting sewage into the creek. So that wasn't all of it, but that was one of the major ones. So that's a place where things can come from either improperly, or if these just fail on their way to the main sewer line, they can just start leaking into groundwater, which can make its way to the creek. Animals, livestock in particular, is another thing that can contribute to water pollution. Also birds. Here's an example from the University of Washington, Bothell, where they have a gigantic crow rookery, and tens of thousands of crows nest there every night. And not surprisingly, where North Creek runs through the UW Bothell, it picks up a lot of bacteria as well. So let's talk about how we measure bacteria. This is an example of the equipment that's being used to monitor the bacteria. So they have a few basic principles, Global Water Watch does. One is do it the same way, same place, same time. They advocate using this simple low-cost method. If you do it the same way using consistent methods and techniques, you can generate credible data. And always, if in doubt, do it again. So in other words, make sure you don't make a mistake when you're doing the technique. If you do it over, so you're not generating incorrect data. So we're monitoring for E. coli and general coliform bacteria, and these are examples of what we're going to be using for our monitoring. When we talk about how we measure bacteria, we measure it in terms of things called colony forming units that are abbreviated CFUs. So this is an example of an incubated petri dish of water and media where the E. coli colonies react with an enzyme and turn blue, and other coliform bacteria's colonies result in a pink color. And so what you do is, we'll talk about how you count them, but essentially you identify the number of those dots that are big enough for you to count and come up with a number of colony forming units. So again, a CFU is a measure of a colony of those cells that's viable enough to form a colony. So in other words, if you just had a tiny dot, you probably wouldn't even see it. But if it's big enough to grow into one of these larger colonies after you incubate it, then you would count that as a colony forming unit. The standard that agencies and people use when they're talking is the amount of CFUs per 100 milliliters of water in your sample. Global Water Watch has some standards that they train us on for what is a risky level or what is not a risky level. So in the Global Water Watch standards, greater than 600 CFU per 100 milliliters is considered unsafe for contact. 200 to 600 is safe for infrequent contact, and less than 200 is generally considered to be safe. Washington State has stricter standards, and so they changed their standards recently and simplified them somewhat, but essentially they came up with less than 100 is considered to be safe, greater than 100 is considered to be risky. We talked about a concept of what's called listing. So a couple concepts here. One is what's called a 303D list. Section 303D is a section of the Federal Clean Water Act. And under that section, a creek or a water body can be identified as being impaired due to a specific pollutant. And that could be bacteria, but it could also be low dissolved oxygen. It could be high temperature. So if somebody collects data, somebody thinks that a creek might have a pollution problem, they collect data that says, okay, the bacteria level is too high or the temperature level is too high, then they'll submit that data to the Department of Ecology, who add it to this 303D list. So if you want to look up which water bodies are polluted with different types of pollutants, you can search for 303D listings for the creek that you're concerned about. Another concept is a thing called a water quality improvement plan. So, for instance, let's take the example of Swamp Creek has a listing, a 303D listing for fecal coliform bacteria. So the city of Kenmore and other agencies that have jurisdiction over Swamp Creek have to have a water quality improvement plan where they say, we're going to put these measures in place to improve the water quality of Swamp Creek. That brings into play another concept, which is the total maximum daily load, which is referred to by the acronym TMDL. So that's essentially what is the maximum level of pollution we can find in this creek. And they're hoping that it will be less than that. But essentially, they're saying, we're going to monitor this water body monthly and see how the level of pollution that we find for this variable compares to what the criteria is in the TMDL. So a creek gets listed on a 303D list. They develop a water quality improvement plan that establishes permissible levels for pollutants. So a little bit technical there, but that's just to give you some vocabulary about water pollution and bacteria. Okay, so how do we actually do the monitoring? So we have some different materials that we're going to use. We're going to use Petri dishes. We're going to use this stuff called Coliscan Easy Gel, which is a medium that essentially it's like food for the bacteria. We're going to use a pipette to collect our sample. We're going to record the air and water temperature with a thermometer, and we'll also record a temperature while it's being incubated. We're going to use an incubator, and there's different designs of incubators that you can use. Global Water Watch teaches building a simple low-cost incubator using essentially a cooler, like a nightlight, and a little cord. We originally started with that model, but we've ultimately moved to using an egg incubator with a thermostat because it's just easier to work with. So you're going to go out and go monitoring. You're going to get your other materials together. You need your data sheet. Once you collect your sample, you can either transport it with not on ice less than 30 minutes before you incubate it, before you plate it and incubate it, or if it's going to be greater than 30 minutes, you need to transport it on ice. So we bring a little ice pack with us in a container. We're going to get our Petri dishes ready, and you'll learn there's a certain way that we basically put tape on them so that you can open it up and then seal it up so that you can put it in the incubator and not get any additional contaminants in it. So we'll pre-label our Petri dishes and our media before we do our sampling. And you can see that there shows 3 different samples. So for each site that we're going to sample, we're going to collect 3 what we call replicates. So 3 different samples, and then those get averaged to come up with your number. So we'll label them to make sure that we're not accidentally putting our 2 samples in the same dish. So we're going to have, for instance, 3 samples here, 3 Petri dishes and 3 samples that correspond with that. So in terms of collecting samples, when you go out to the site, you're going to unwrap a sterile pipette, collect the sample. You want to be facing upstream so that you're not interfering with the water that you're sampling. And you want to be below the surface of the water, but not on touching the bottom. You'll collect the sample and put it into the media bottle and then recap it. Then you bring it back for plating. And I'm going to show you guys a video on how to do this.
Welcome to the Alabama Water Watch instructional video series. This video will show the proper method for collecting water samples used to determine the number of bacteria in the water body. This is the first step in a three-step process. The second and third steps, plating the samples and determining results, are covered in their own separate videos. For each site being tested, you will need a cooler with ice, 3 media bottles at air temperature, a sterile pipette, and a thermometer. Label your media bottles by site and replicate. If you sample multiple sites, be sure to use separate data sheets or keep record of monitoring site and bottle numbers that are grouped in sets of 3 replicates. For example, if you have 2 sites, you can number the replicates at the first site as 1A, 1B, and 1C, and the replicates at the second site as 2A, 2B, and 2C. Upon arrival at the site, measure the air and water temperatures. For instruction on how to measure these temperatures, please watch the video called Water Chemistry, Air and Water Temperature. Once you are ready to sample, unwrap a sterile pipette from the bulb end and avoid touching the tip of the pipette with anything except the sample water. Fill the pipette with sample water. Be careful to not disturb the bottom of the stream. Carefully squeeze the bulb and expel excess water to the desired sample volume. 1 milliliter sample volume is recommended and is usually sufficient. Be sure to use the same volume for each of the 3 replicates and record volumes on data forms. Remove the cap from the media bottle and hold it without touching the inside. Squirt the sample water into the bottle of liquid media without placing your hand or face over the open bottle. Cap the bottle securely and place inside the cooler. Repeat these steps until all 3 media bottles are inoculated with water from the same spot. Keep in mind that media must be plated within 3 hours of collecting the sample and should remain on ice and out of direct sunlight at all times. Thanks for watching. If you have any comments or questions, give us a shout.
All right, you guys have any questions so far? No? All right. Dave, you got anything to add?
I've got a question, Eric.
Sure.
Where do you get this stuff? Is there like a kit you have to buy, or do you just go out and get it on your own?
So if you're monitoring in a program that's associated with us, then we'll provide you with the supplies. Yeah, I didn't mention that in the very beginning, but we are operating under a grant right now from King County Waterworks program. And so that is paying for our water monitoring supplies, like our bacteria monitoring supplies.
Cool.
Okay, so that was collecting the sample. So the next part that you do is what's called plating the sample. So we've gone to the stream, we've collected our sample, we put it in the bottle. One thing we didn't mention, they talked about the sample size. We'll get into sample math a little bit in the future, or in the next couple slides. But so we bring our sample back. Now we're going to do what's called plating. So we're essentially going to pour the sample into the Petri dish, and we're kind of moving to a slight change from what they used to do. We used to plate the samples, let it gel, because they're pre-treated with something that will cause the sample to gel up, then put it in the incubator. Now we're essentially just pouring it, plating it directly in the incubator. So we do this plating process with a dish in the incubator. So you'll open it up, pour the media in, swirl it to cover the bottom, but not too much because you don't want it going way up the sides. That'll make it difficult to count colonies. Like I said, we used to let it gel first. Now we just plate it right in the incubator and then incubate it. So I'm gonna show you guys a video on how to do plating. Oops, not that one. Plating.
Welcome to the Alabama Water Watch instructional video series. This video will show the proper method for plating water samples used to determine the number of bacterial colonies in a water body. This is the second step in a 3-step process. The first and third steps, collecting the samples and determining results, are covered in their own separate videos. Prepare your petri dishes by taping the top, the larger piece, to the bottom, the smaller piece, on one side of the dish. Keep the dish closed as much as possible to avoid contamination. Label the plate lids with labels that correspond to the inoculated media bottles in data form. Lift the top of the petri dish just enough to pour the contents of its corresponding media bottle into the plate. Do not slosh the media onto the sides or lid of the plate. Record the time you plated your sample on your data form next to incubation start time. Be sure to shield the sample plates from direct light. Leave the plates undisturbed until the media has gelled, usually about 1 to 1.5 hours. After the media has gelled, use another piece of tape to secure the untaped side of the dish. To prevent condensation from dripping onto the plates from the lids, turn the plates over and place them in the incubator. Incubate plates between 29 and 37 degrees Celsius for 30 to 48 hours. Thanks for watching. If you have any comments or questions, give us a shout.
Okay, any questions on plating?
So I assume if we're placing them directly in the incubator rather than letting them gel, we're skipping the flipping part?
Yeah. Yep.
Right.
Yeah, it's just, it has to do with somehow over time the petri dish production has changed or something, so that they weren't gelling up as well. And maybe it has to do with people using larger samples, because you can use anywhere from a 1 to a 5 milliliter sample. Typically we use like anywhere from about 1 to 3 milliliters, but if you have a larger sample, it takes longer to gel. But it just proved to be kind of a unnecessary step, and sometimes it didn't gel in the exact amount of time, and so it just ultimately Global Water Watch issued a update to their procedures and said go ahead and plate it directly in the incubator.
And do we have more specific, well, 2 questions. First of all, do we make our own incubators, or do you provide them?
So we provide the incubators, and actually, hang on a second, I got one right here. So the incubator that we use looks like this. So it's got it basically is this stuff taped on it it comes with Windows. It's an egg incubator, but basically it's got a thermostat. And so we just plug this thing in, open it up, and put our dishes inside of it. And then what's nice about this is you can program in, and we pre-program these with a set temperature, so it maintains the temperature like it's supposed to for the whole time. With our homemade incubators, we used to have an issue with, if one person would, if we transferred it from person to person, one person would put it in the garage, another person put it inside their house, and we might get our temperatures out of range. And so just it lets our technique be more consistent.
And the incubation time is fairly broad, between 30 and 48 hours. Are we more specific about that?
No, essentially you just have to give enough time for the colonies to grow. I think if you go beyond that, you might get like a lot colony overgrowth, or it might be hard to count the separate colonies distinctly. So,
okay,
yeah, and that gives you a window depending on when you do your plating. if you do it it just lets you set the clock. So if you do it in the afternoon, you wouldn't necessarily have to then do your counting in the middle of the night, if it timed out a certain way.
Fair. Yeah. Thanks.
Okay.
Yeah, and actually, if you do run it a lot longer, it's usually fine. So I think what happens is the medium dries out, and once it's dry, things stop growing. So you really don't have that overgrowth problem unless you like leave it in a bathroom where people are taking showers all the time and the humidity is really high. And then I guess the other thing that can happen if you let it run a little long is you start getting other things like mold growing on there that can obscure things a bit. But I've had some that I've run for a month and they basically look the same after a month as they do after 30 hours. So running too long doesn't make a difference. And then running too short, the colonies are smaller. So we have a size criterion for what's big enough to count and what isn't. And some of the ones that would grow to be big enough to count, aren't there by 30 hours, but by the time you get to 40, they're clearly big enough to count.
Dave, did you intentionally let those go for a month, or was that an accident?
It's intentional, and I've got some that instead of killing them the way we're supposed to, I've let them continue to run at room temperature for over a year, and they still aren't changing very much.
All right. Well, we're not advocating that.
The correct procedure is to kill it with a spoon of bleach after you've counted it, and that way you don't have to worry about infecting yourself if you got a dangerous strain that you happen to colonize. But I'm a biologist, so I have different curiosities than other people might have. And I've also been known to let whales collect my samples for me. So they exhale and blow a bunch of water into the air, and then I'll collect the droplets as they settle down and culture those.
Okay, so the next step, so we collected our samples, we plated and incubated. Then our next step is going to be, we're going to determine the results. So I've got another little video to show you guys on how to do that.
Welcome to the Alabama Water Watch instructional video series. This video will show the proper method for determining the results of incubated water samples. This is the third step in a three-step process. The first and second steps, collecting the samples and plating the samples, are covered in their own separate videos. Remove plates from the incubator, being careful not to spill any liquid. Be sure to record the incubation period, which is optimally between 30 and 34 hours. When counting the number of colonies, it is helpful to use a white sheet of paper to split the plates into quadrants. Count all blue to purple colonies on each plate as E. coli. Do not count very small or pinpoint colonies smaller than the period at the end of a sentence. Count or estimate the number of all pink to dark red colonies on each plate as other coliforms. Estimates of more than 100 colonies should be reported as abundant, and estimates of more than 200 colonies should be reported as too numerous to count. This sample should be recorded as too numerous to count. If colonies appear as a ring around the edge of the plate or as one large blob, count as one colony. Record both the number of E. coli and other coliforms on the data form. The bacterial concentration in 100 milliliters will automatically be calculated in the Alabama Water Watch database. To calculate the bacterial concentration by hand, divide the number of E. coli colonies counted by the sample volume, then multiply by 100. Kill the bacteria by pouring about a teaspoon of undiluted bleach into the dish. Let stand for about 15 minutes, then dispose of with household garbage. Thanks for watching. If you have any comments or questions, give us a shout. Welcome to the Alabama Water Watch instructional video series.
So just to clarify a couple things on there. One is they said 30 to 34 hours, so that was just a mistake. And then as far as cleanup, Dave was talking about the bleach. I've had some other people ask me if it's just okay, if you're not going to reuse the Petri dishes, can you just put them in a Ziploc bag and dispose of it, landfill it? And I don't really see a problem with that. That's not the official technique which is taught, but I think that you could also dispose of it that way. But the way that they teach to do it is to open it back up, pour some bleach in it, let that sit in there for a little period of time, and then you can basically dispose that, or you can dump that. It'll slide right out. You can just dump that down the toilet and get rid of it that way. I don't know, Dave, do you have anything you want to add about that?
Yeah, if you don't kill it first, then you run the risk that something is going to eat it. And again, if you have a dangerous strain, which is rare in E. coli, but it does happen, a mouse or a rat or something is going to get a very large dose that it can start dropping in the environment for other critters to pick up, or to wash into a stream where people can pick it up. So it's probably best to kill it. But as I said, I've let some of my colonies run over a year and I haven't gotten sick from it. So it's sort of a low risk thing if you don't bother to kill it.
All right, so we're going to talk about how to count bacteria. So we've done our incubation, we pulled it out of the incubator, and now we're counting. So how many, let me know, you guys, how many E. coli colonies do you see in there? So those would be the blue colored ones.
Looks like 7.
Yeah, I confirm.
Also what I see.
Okay. So 7. So then so our sample volume was 1 milliliter. So if there's 7 in 1 milliliter, what's the number in 100 milliliter?
What is 700?
Sounds like a Jeopardy. So there's the math, and correct, 700. So then the number of other coliform really depends on if you can count them or not. And another thing that you can do is you can see that this has been divided up into 4. You can divide, I essentially take a Petri dish lid and I've made it a grid into 8. So I could divide it into 8. So I could count the amount of other coliform that are in 1/8 and then multiply it out. So you go through and count all those colonies. You don't count the ones that are super small. So in this case, it counted 45 and 1/8. So our other coliform is 360, which would be 36,000 in 100 milliliters. We don't really worry about that number. That's kind of just a reference number. We record it, but the E. coli is really the key number that we're concerned about. The other number would be more like, it'd be unusual if you found no other coliform when you had a bunch of E. coli. So that might tell you there was something going on with your sample or with your materials. Going back just so to interpret this, so would 700 milliliters or 700 CFU in 100 milliliters, 700 colony forming units, would that be a good source for drinking water? You guys remember what the level is?
I sure hope not.
Yeah, so our cutoff would be 100. And really, I mean, even 100, I don't know if you'd want to drink it, nor would you want to swim in it. So here's the standards for Global Water Watch. Washington state standards are even stricter still. So here's another example. So this is kind of like a math example. So in this case, our sample volume was 5 milliliters. And how many colonies do we have here?
7 still.
Yeah, it looks like 7. So if we had 7, our formula is the number of colonies divided by the volume of our sample times 100. So our number of E. coli would be 140 CFU. So how does that compare to our standards? Do you guys remember?
Still wouldn't drink it.
Yeah. Still wouldn't drink it. By Washington state standards, you really wouldn't want to be in it, but it's less risky. It's not super risky.
Yeah, so I'd probably canoe or kayak in 140, but I wouldn't swim in it and definitely would not drink it.
Yeah. So these are just some other exercises we could do with the math, but essentially you guys get the idea of if you vary the sample size, you have to adjust your math to come up with the number of colony forming units per 100 milliliters. So once we've done our counts, then we record it on this data sheet. And so you can see all the different things that we record on the data sheet, information about the monitor or the site, the water body conditions, air and water temperature, what was our sample volume, how long did we incubate it. The incubation time is what time did you start incubating it, and we put that down as the time when you plate the sample as your incubation time. And what was the temperature, how long. The media does expire, so we always check before we monitor to make sure it hasn't expired, and we record the expiration date on the form. Plate it on site. We generally don't plate it on site, and you may or may not transport on ice. And then there's a place to record the E. coli and the other coliform. So then you do cleanup, which we kind of talked about. And then ultimately you're gonna do something with your data. So you can kind of interpret, think about what you're finding and what does it mean. So if you're finding it at a place that's commonly used for recreation, maybe you wanna share some information with, if it's a public site, maybe you wanna find out what agency controls that site and say, hey, maybe you guys could post a warning sign, or would you like us to post a sign? Or you may find that, there's a high level, like for instance, with the Thornton Creek example, they looked at multiple sites within the watershed. They were trying to pinpoint a source. And so they used that to find their source. And they zeroed in after monitoring multiple sites to monitoring in a particular, closer to where they were finding the high levels. And so you can use, this is an example of the graph off of the Global Water Watch site. So you can use this to present data. You can also turn it into a poster. So here's something that Lake Forest Park stream keepers came up with, showing kind of what it normally looks like when they sample, and then what it looks like after a heavy rain. And so that's a great graphic example. And I think they used this when they were having some of the, I think they called them Green Days events at the town center in Lake Forest Park. You can do something like what I talked about with the Thornton Creek example, where you can collect data simultaneously if you're trying to pinpoint a source within a watershed or a particular stream. Here's an example where at Auburn University, they found super high levels of contamination at a pond in a thing called the Memory Garden. And so they put up a sign warning people about it. So there's different things that you can do with your data. Here's an example of the Global Water Watch program working in Kenya. They had people take the samples and they drew a watershed map on the floor. They laid out all their samples, and then they transferred that into a larger scale watershed map so they could identify where they had water quality problems and where they didn't with regard to bacteria. Another way that this is used, there's some groups called waterkeeper groups, and they have a thing called the swim guide, which they identify bacteria levels at swimming beaches. And so this is another thing that can be done. You know, in some cases, oftentimes swimming beaches like around here are monitored by a health department, but there are instances where a volunteer group might do that monitoring, and the swim guide is an example of that. So this would be off the swim guide. They could say, okay, you know, is this a place where I typically want to go swimming or not? Here's an example from Global Water Watch Mexico, where they just had an informational poster they put showing, comparing different sites and showing what the results were in the different sites. All right, and these are the folks that helped us get our program going and have been funding our program.